So we're going to talk very briefly about tumor suppression and apoptosis. So that's just, a quick picture, normal cells and cancer cells. You can see cancer, for those who wouldn't know, cancer is just an unchecked growth of normal cells. A bunch of different ways how cells can go haywire. We're not going to focus on that too much in this lecture, but it is important for this discussion. So tumors are often caused by mutations in genes that regulate cell signaling. Cell proliferation is regulated very delicately by each cell. Like we said in the beginning of the lecture, with that huge picture with all of those different, signal pathways. But a disruption in any of those signal pathways can result in unchecked and potentially malignant tissue growth. And some receptors may even fire independently of extracellular ligand binding. So, it's usually mutations or, we could talk for hours about how cancer starts, the different pathways, the checklist of things a cell has to go through. But receptor tyrosine kinases are one of the types of receptors that are very often implicated in cancer. And I just put another graphic at the bottom so you can take a look at that. The difference between ligand-dependent firing and ligand-independent firing is right here. So the one on the left right over here is only going to fire, and it's only going to send out proliferative signals to the rest of the cell if there's that GF growth factor is attached to this receptor tyrosine kinase. Sometimes when these receptors are mutated and the cell starts pumping out a bunch of them, you could see over here, one of a couple of things could happen. It could either stay close, or there's no actual extracellular domain on it. So there's no extracellular domain. So these are just dimerized constitutively, and they're just constantly sending out these signals, which is an issue because, again, unchecked cell growth leads to tumors. It could lead to metastasis, and it could lead to death. So your cells have developed a way to monitor cell growth and destroy any cells that are proliferating too fast. And then they're in a bunch of different ways. They have just like natural cell checklists. Your immune system plays a role as well. But what we're going to talk about today, one of the more important proteins that your body makes, is known as P53, and that is sort of the famous human tumor suppressor protein. So the gene that encodes for P53 is a gene that specifically encodes this protein to check tumors, to keep tumors and unregulated cell growth in check. So P53 protein levels in a damaged cell will rise and eventually trigger a signal cascade. So if we look to the right over here, if you look at the P53 response at the bottom, whenever there's like an increase in P53 production, that obviously means that there's a problem with the cell. So it will try one of those, one of those 5 things. The cell could enter senescence. Cell cycle could be arrested until maybe the cell tries to do DNA repair, or it tries to figure out what's going on. The cell could also differentiate, but a lot of times when there's nothing left for the cell to do, when it's not contributing, and it could even, and the body recognizes that it could be a threat to the surrounding tissue, it undergoes apoptosis. And that is, again, a very popular signal cascade that you may see on the MCAT, which we're going to talk about in a couple of minutes as well. We'll talk about right now, actually. So the apoptotic signal cascade, apoptosis is the pathway for programmed cell death. And it is a last resort strategy, which destroys the cell from the inside out so as not to damage any surrounding cells. And that's super important because there are a bunch of different ways in which cells can die. Necrosis, autophagy, there are a bunch of different ways they can die, but apoptosis is specifically programmed for cell death. So it's just the body's way of getting rid of cells that we don't need anymore, and it does it in a very safe way so that it doesn't damage any other healthy cells that are around it. So it's sort of a very quiet fizzle out rather than a huge burst, as like a cell that is lysing would be. But during apoptosis, the cell first begins to shrink. As the cell shrinks, the cytoskeleton is disassembled. The cell starts to bleb, is what they call it, and it starts to get sort of bubbly, and it starts to bud out because the cytoskeleton isn't really holding anything together anymore. So the nuclear membrane starts to break down, the genome starts to become fragmented, and then the tiny remains are broken down and recycled by phagocytic cells. So if we look at the bottom, exactly what I just said. There's a normal cell, the cell shrinks, the chromatin starts to condense. You can see over here, the membrane starts blebbing, the organelles start to disintegrate. There are caspases, which we're going to talk about in a second, that are just flying around the cell. They're destroying the proteins. Everything's being broken down. The cytoplasm is blebbing off, and it's just breaking down into tiny little remnants of the cell that it used to be. So then these tiny little apoptotic bodies form, which they're basically just little pouches of broken up protein. And then they're digested, and they're usually, if there's anything that's recyclable or reusable, the cells will do so. But that's basically what apoptosis is. And as far as the actual signal cascade, apoptosis is carried out by that cascade, which involves enzymes that are known as caspases. So caspases are a class of proteases. Remember, proteases cleave proteins. And the reason why they're called caspase is because they use a cysteine to cleave target proteins at an aspartic acid residue. So you can see that C comes from cysteine, the ASP comes from aspartic acid. So that's where the name caspase comes from. And there are actually two types of caspases. So the first one that we'll talk about are called initiator caspases, and obviously the two of these have two different roles. The initiator caspases are the ones that are first activated by whatever the first death signal is. So for example, the example that we've been using is P53. Whenever the cell sees that there's stuff going on that shouldn't be going on, maybe that cell's becoming a little bit malignant, maybe the P53 levels are going to increase, and that signal transduction is going to take place, and that's going to result in the activation of these initiator caspases. So these initiator caspases are going around, and they are going to activate more initiator caspases. So they activate themselves, and they also activate effector caspases. So that's also how the signal is being amplified. If you initiate one initiator caspase, for example, it's not just going to be one initiator caspase starting all of these effectors. It's going to start initiating other initiators as well. So that is where the signal amplitude comes in. So then it just grows, and it's a sort of snowball effect. And the effector caspases that are activated circulate the cell, and they break down proteins to trigger apoptosis, like we just mentioned before. Okay, so here's the final review question. Okay, so which of the following statements is true about caspase cascades? So the answer to this question is C. And we could read all four of them, and we'll just go through them individually. So Roman numeral 1 says, initiator caspases activate effector caspases. So the question, remember, is asking which of the statements is true. So initiator caspases do activate effector caspases. So that is a true statement. Roman numeral number 2, initiator caspases activate other initiator caspases, which is also true. So since we know that 1 and 2 are correct, we can actually automatically eliminate A and B because 2 is not a part of their answer. So when we look at Roman numeral number 3, again up here, the effector caspases activate initiator caspases. That's not true. The effector caspases are the ones that are actually carrying out the catalytic effects of apoptosis. So they're actually going around and they're cleaving all of these proteins. The initiator caspases activate effector caspases, but it does not happen the other way around. So 3 is not correct. And then from there, you can eliminate D, and that leaves us with C. And obviously 4 is correct as well. Caspases cleave proteins at the aspartic acid residue. So that's true as well.